Today I'll be describing a means for eliminating a single bacterial pathogen from a complex microbiota. In this case, we are looking at Helicobacter pylori, and we'll be doing so using a very convenient method using engineered probiotics. Helicobacter pylori is actually a very important pathogen. It is the cause of 78% of all gastric cancer cases. It's listed as a group 1 carcinogen. So many groups today are interested in possible bacterial links to cancer development, but H. pylori really is the model. It's not just wondering if this can happen, but it actually has been shown to be the cause. So it's an important pathogen. 750,000 deaths per year globally by gastric cancer. But interestingly, H. pylori infects 43% of the global human population. So the vast majority of people are non-symptomatic. The control of H. pylori is now dominated by antibiotic treatment, and especially by multi-drug antibiotic treatment. But even with the multi-drug approach, there's high resistant frequencies, especially in East Asia, occurring. It is, of course, a problem with all bacterial pathogens, but especially so with H. pylori. So the treatment success rate is actually dropping now, and there's a lot of concern about this. Antibiotic stewardship would be a great solution, but unfortunately, inexpensive over-the-counter antibiotics are common in South and East Asia. So it makes stewardship difficult. So if we were going to design an improved H. pylori control agent, what would be the main goals? Well, unfortunately, we have to be as effective as antibiotics, and excluding the problems with drug resistance, antibiotics are very effective. Even worse, we have to be as inexpensive as antibiotics, considering in East and South Asia, there are a lot of countries and regions which are impoverished. And so antibiotics provide the only method for having any realistic control measures for the large part of the population. It would be also very important to avoid the development of drug resistance, because this is the reason we're looking for something other than antibiotics in the first place. Another great goal would be to avoid broad spectrum action. And the reason I'm saying that is that with broad spectrum action, even though you get all the pathogens, you get everybody else in the gastrointestinal microbiota. And so that flattening, that destruction of the microbiota brings many problems, many health problems. Not to least to mention would be the introduction of new pathogens into a flattened playing field after the antibiotics destroy everything else. So the approach we're taking is the guide peptide approach. And you may not have thought about this before, but of course, guide peptides are common in many different areas of biotechnology. In this case, we are taking a guide peptide that binds, and I've written this in blue in the diagram at the right, a guide peptide that binds to a target protein in green, which is residing on the bacterial membrane. So it's a bacterial protein, which is on the surface of a bacterial membrane, and we're using a guide to be able to identify that and bind to it. This guide peptide is fused to an antimicrobial peptide. And the guide peptide brings the antimicrobial peptide close enough to the bacterial membrane where it can attack the membrane and kill the bacterium. So we call this guided antimicrobial peptides, or GAMPs, and that's the way that works. Now, the specificity is in part due to an increased toxicity of the guided antimicrobial peptide, sometimes, but not always. Where the specificity occurs even more is somehow the guide interferes with the antimicrobial peptide, attacking other bacteria that are not targeted. These I call off-target bacteria. So the guide provides a specificity by binding more strongly to the targeted bacterium, but also laying off and not really harming the off-target bacteria. So the benefits with guided AMPs is they have a very narrow spectrum. So this would preserve the microbiota diversity in therapy treatments. But there are a couple other benefits that are not as obvious, but our lab is really looking into. And that is when you have the question of a targeted therapy, you're going to expect to get some sort of resistance developing via evolution. So let's think about how that resistance evolution would occur with this sort of system. Well, the evolution would occur against the guide-target interaction. In other words, the fusion, guide peptide fused to the AMP and the targeted protein. So the targeted protein is produced by the bacterium, and the targeted protein should evolve in order to evade binding by the guide. But what if the targeted protein was a virulence factor of the bacterial pathogen? That means that the virulence factor would need to evolve in order for the bacterium to evade being targeted by this therapy. If the virulence factor evolves, then it might be possible to use this peptide in order to cause the population of pathogens to evolve towards avirulence. A second interesting benefit, potential benefit, is guide peptides are not that difficult to make in comparison to new antibiotics or even new antibodies, monoclonal antibodies. So it would be possible to make a cocktail of guided AMPs, and let's say that you had 3 different guided AMPs. Each one of those would have a different guide with a different target. So it might target a different surface protein. If this were the case, then we could have a multi-drug type of approach with each one having a different mode of action. And this might contribute to slowing down or even stopping resistance evolution. So 3 possible benefits with guided AMPs. Now we'll look at the results of binding of the specific guide that we have used for the study I'm describing today. This guide we call MM1. It is derived from multimirin 1, which is a receptor for H. pylori. Specifically, it is bound by a virulence factor of H. pylori, VAC-A. So VAC-A binds to multimirin 1, specifically at this sequence, which is called MM1. Now, this study was done previously by another group, and it was done against purified VAC-A protein. So we used this, this guide peptide because it was there in the literature, and we bound it, first of all, to GFP to see how well it binds, if it binds specifically to H. pylori. And you can see in figure A here that GFP, and this is flow cytometry results, GFP does bind to H. pylori slightly, but when you have GFP, which is fused to MM1, the guide, it binds much more strongly. Now, if we use these same 2 proteins, GFP unadorned and then GFP guided, we find that there's no difference between the unadorned GFP and the MM1 guided GFP. And the same could be said for looking at E. coli. So for the off-targets, lactobacillus and E. coli, we get no increase in binding with a guided GFP, but against H. pylori, we get a big increase. So it looks like we have specificity. Now, one thing I want to note in the interests of being very careful scientists, we actually went and obtained a delta, well, it's not a delta VAC-A, but it's a VAC-A knockdown mutant of H. pylori, and we also built a knockdown mutant of VAC-A H. pylori with our own hands. And we tested both of these. And the interesting thing here is that we got an increase in toxicity with the guided GFP against the VAC-A knockdown compared to just unadorned GFP. So it appears that the specificity does not actually require VAC-A, that I would suppose there must be some other protein or proteins which have co-evolved in H. pylori to be able to bind this receptor. So just to make sure that we have things all set, we went out and looked at more off-target bacteria. And as you can see, we got the same binding of GFP and MM1 GFP for Pseudomonas, Alcaligenis, Staphylococcus, and I don't have this graph, I didn't make it into the slides, but Acinetobacter as well. So this MM1 guide peptide binds only to H. pylori and not to the other 6 off targets. So by whatever mechanism, it's a highly specific guide peptide. Well, that's great. We've got a guided antimicrobial peptide, but there are problems with using peptides to try to control a gastrointestinal pathogenic bacterium. And for one thing, we have to remember peptides are actually digested by the stomach. So to solve this, we have used a probiotic delivery system using specifically the probiotic bacterium Lactococcus lactis. Lactococcus lactis is resident in the human stomach, so that means that it can bring the peptide right to the stomach, make it all the way through all that digestive hurdle, and then deliver it right to where the H. pylori is located. Another problem with using peptides for as a replacement for antibiotics against gastrointestinal disease is that peptides are just too expensive for widespread use, especially in low-income areas in the East Asia and South Asia. Well, Lactococcus lactis is very attractive because it is a food-grade dairy component. Lactococcus lactis has been used by humans for thousands of years with dairy components, and it is registered as a GRAS, or generally regarded as safe bacterium. And so this would be, it can be scaled up very easily, and it's very inexpensive, and it's generally accepted by the public. So this would be a great solution. Now, we are making an engineered probiotic, and that puts another wrinkle in things, but it is an interesting thing that there is a company right now, Z-Biotics, which is commercially selling on the internet engineered probiotics for another application, and you can look at their website. They are not FDA approved, but they are FDA registered, and that seems to be working. So, how are we going to engineer the Lactococcus lactis? Well, as with lots of other bacteria that need to be engineered, we have constructed a shuttle vector, and in this shuttle vector, we have the origin of replication for E. coli and the origin of replication for L. lactis, and separate antibiotic resistance genes for each one. So we go ahead and do our engineering in E. coli, and then we electroporate into L. lactis, take a plasmid prep from E. coli, and then electroporate that into L. lactis. Then once it's in L. lactis, we have a secretion system working here with a signal peptide, and we can secrete the guided antimicrobial peptide. And here are some results of in vitro co-culture. So this is an emulation of what might happen in a mouse or human stomach. And so we culture the L. lactis, and you can see at the bottom of this figure, we have all of the different increasing L. lactis dosages that, if you could call it a co-culture, a dosage. And then we have on the y-axis, we have the recorded amounts of H. pylori. So we start off with 100,000 CFUs of H. pylori, and then we watch how that, we look at, it's not over time, but we look at the effect of co-culturing this with differing amounts of L. lactis. And then 24 hours later, we go ahead and register how much H. pylori has survived. And first of all, let's go ahead and look at the green graphs down on the bottom section. And these are L. lactis, which contain just an empty vector and are expressing no AMPs. And this is a log scale, mind you, so we do see some decrease at the higher amounts of L. lactis. We do some, it does to dampen the amount of H. pylori, which survives somewhat. But when we look at the L. lactis, which is secreting either unguided AMPs, that would be in blue, or guided AMPs, we see a big decrease in the amount of H. pylori which survive the differing amounts of L. lactis. And so notice on the left for the target bacterium, H. pylori, we can see that we have the unguided AMP is killing H. pylori, but it is better when you have a larger amount of L. lactis expressing, of course, a larger amount of AMP. That's where you finally get some kill. But when we have the guided version of the AMP specific to H. pylori, and when the L. lactis is expressing that, and that is in red, you see a big difference. And this is a log scale, so we're seeing 80 to 100-fold differences in the amount of kill we're getting at the mid-level amounts of probiotic that's being added. And we went back and repeated that experiment using purified peptides, and we got similar results. So it does seem that the presence of the guide increases the toxicity greatly of the antimicrobial peptide. And by the way, we tested this. This is just the test for elitacerin. We did tests with 2 other antimicrobial peptides as well. But when we look at off targets, in this case, we're just looking at E. coli and lactobacillus, it's flipped. So now the unguided AMP is on the bottom, and of course that kills okay. Now it levels off just because there's a certain level of detection that we can use for E. coli and lactobacillus with using qPCR, and we actually use qPCR with this and we coordinated that with CFUs. So that's why you see a leveling off there. But when you add the guided AMP expressed by lactobacillus, this hangs way back. And so we're seeing a 20 to 100-fold less killing when we have the guide present. So with the guide, increased toxicity against the target. Without the guide, decreased toxicity against the off targets. So now on to the mouse model. So what we did here is we, of course, used the TKR shuttle vector, and that was we transformed that into L. lactis, and then L. lactis was supplied to the mouse via oral gavage, and then the guided AMP was secreted on site by the L. lactis, and then this guided AMP was able to find the H. pylori because it specifically binds H. pylori and to kill it. So let's see some of the data. Well, before we see the data, let's look at the experimental design. We did both a therapeutic experiment and a prophylactic experiment. We used a lot of mice in this study. And this study, by the way, is in press now with microbiology spectrum that's put out by American Society of American Society for Microbiology. That's one of their imprints. And so the therapeutic experiment, we have to get the H. pylori going and having a very strong infection. And so what we do is we actually went 4 days in a row by oral gavage. We supplied H. pylori until we got a very strong infection in the mice. We gave the mouse a break for 1 day, and then and the 5th day we gave the Lactococcus lactis. And so then we waited for 2 days, and then we sampled using a reverse oral gavage, and that's by flushing the stomach with a buffer and then drawing that stomach out, drawing the buffer out, and so we don't have to sacrifice any mice. And then we did that on the 10th day as well. And mind you, we did a reverse oral gavage on day 0 as well, before we added the H. pylori. So we have the values for what we have, and this is for all of our mice. We have the 0-day values, and getting an idea of what the mouse microbiota looks like without H. pylori. And then we also did a reverse oral gavage just before we added the L. lactis on day 5. So we also have a peek at what it looks like in these mice stomachs after they've had 5 days of H. pylori. So those are 2 values that I'll be coming back to later no H. pylori, and then with H. pylori. And those are going to form bookends as we establish a microbial um, um, a dystrophy index. So we're going to come back to that later. Dysbiosis index. So that's the therapeutic experiment. The prophylactic experiment, we give it L-lactis at the beginning, and that's just a single dose, just as in the therapeutic experiment, it's just a single dose. And then we let the L-lactis grow out a little bit, and then starting on day 3, we supply the H. pylori 4 days in a row, and then we sample. And of course, we sample at day 0, we sample at day 3 before we add the H. pylori, and then we sample at day 8 and also day 10. So let's just look at the therapeutic results for right now. And you can see at day 0, we have, this is a peek at what it looks like with the amount of H. pylori in mice at day 0. And there is no H. pylori in mice at day 0. And so then we have at day 5, when we, just before we add the probiotic inoculation, we have lots of H. pylori. Then day 8, 3 days after probiotic, day 5 days after probiotic. And let's just look at day 5 because I have this all labeled. And we have the no therapeutic. We have an awful lot of H. pylori represented here. And then with the native probiotic, that is to say, with just the empty vector L-lactis, we we do have some decrease. So the actually the L-lactis did lead to a somewhat of a decrease in H. pylori. Then we have the antibiotic, and we certainly have a decrease with using the antibiotic. But then we have the AMP probiotics, and all of these take us all the way back down to the baseline of having no H. pylori at all at day 10, 5 days after a single dose of probiotics. And we beat even the antibiotic. And note also that in just being able to get rid of H. pylori, the guided AMPs worked, the unguided AMPs worked just as well as the guided AMPs. Now, on to the prophylactic results. Here, at day 0, of course, we have no H. pylori. At day 3, we've had 3 days of probiotics, but we're just about ready to add the H. pylori challenge. And again, we have no H. pylori because we're just about to add it. And then on day 8, this is 5 days post-challenge with H. pylori. Day 10, 7 days post-challenge with H. pylori. And let's go ahead and look at that. I have that labeled. We have no therapeutic. The H. pylori has a nice infection when it's not interfered with. When we have the native probiotic, again, that works as a prophylactic as well to some extent. And I have that labeled as antibiotic. Sorry, that's wrong. That's just plain elitacerin, so ignore that. But all these ones in the bottom are the AMP probiotics, and we have a little bit of H. pylori present. And this is a linear scale, so it's we have just a little bit of AMPs present, but not a lot. And so considering this is 10 days after a single dose of probiotic, I think we're doing pretty well as a prophylactic. So in summary, we're able to eliminate H. pylori from mouse stomach within 5 days with a single dose of probiotic, and we do a little bit better than antibiotic treatment. The prophylactic protection lasts even 10 days after a single dose, and GAMPs and AMPs worked with equal efficacy. So now if we look at the microbial diversity of therapeutic study, and for this, what we're doing is we're just looking at all of the different, getting a count of how many species we're able to measure here, or how many genera we're able to measure here in the gut. So this is just a raw count, the number of genera we're able to measure. And this is using 16S sequencing. So you can see if we have, in the last column on the right, if we have only H. pylori, then after 10 days of only H. pylori, this is the therapeutic study we're looking at, we have a big decrease in the number of species we can record. If we have the antibiotic, we do indeed, we showed previously, we're able to kill the H. pylori, but you notice the antibiotic kills everybody else. So we're actually have a lower number of species than even H. pylori all by itself. And interestingly, with all of the probiotic treatments, we have a rebound effect So going from day 5 up to day 10, we have a rebound in the number of species that we are recording by 16S sequencing, rRNA sequencing, for both the AMP L lactis, the GAMP L lactis, and empty L lactis. And when we look at the prophylactic study, though, it's interesting because in this case, we have the presence of the probiotic for a full 10 days. And here we actually begin to see some differences. We have no antibiotic in this study, but if we have only H. pylori, of course, it goes down very quickly. But if we have the empty L lactis, we do see a rebound, but not as strong as a rebound as we saw before. And if we have the L lactis expressing the AMP, we actually, things don't look quite as good as they did in the therapeutic study. But when we look at the guided AMP with L lactis, we actually see a very strong rebound with the guided AMP. So it's interesting. This is 10 days of that probiotic sitting in there, and this is the effect that we get. So we're very eager to take much longer term studies and see if the guided AMP really is very different and superior to the other probiotic treatments. So in summary, we have of this section, we have H. pylori had a devastating effect on diversity. Antibiotic treatment did not prevent the devastation, even though it was effective at killing H. pylori. It just kills everything else. The probiotic treatments provide a bounce back, all of them do, but if you let the probiotic stay there for 10 days, we begin to see some differential effects. So we wanted to go beyond just counting how many 150 or 170 or whatever genera that we were recording with 16S rRNA sequencing. So we came up with this idea of a microbial dysbiosis index. And what this is we looked at we had samples in our whole total sample set of what the mouse stomach microbiota looked like before we added any treatment or any H. pylori. And then we had samples where we had introduced H. pylori and let it go for 5 days and without any treatments. And so we looked at the change in relative abundance from 0 to 5 days of H. pylori at those samples. And in other words, what is the effect of H. pylori? And what we saw is that there were 8 genera which tended to decrease when you have H. pylori infection. And they were especially hit hard. And then we had 2 genera which tended to increase and actually increase greatly. They thrived after H. pylori infection. So these seem to be the key species we wanted to focus in on. And so we created a microbial dysbiosis index, and this is the sum of the abundance of the red ones that thrived, divided by the sum of the abundance of the blue ones that did poorly, and just taking a log of that. And that's our MDI. And so what we did here is just the standard PCA of taxonomic distances between all of the samples we took in our mouse study. So we had a lot of samples that we took in our mouse study. And so this just shows you how different they are. And you can see they're clearly separating into this one on the far extreme here on the left. And then we have a big band going up and down on the right. Then we went back and we did some coloring, and we identified within each one of these samples what the MDI was for that particular sample. And you can see that the MDIs all cluster in a certain taxonomic assemblage. So if you look at all 150 or 170 species that we looked at, and we separated those by taxonomic distances, the MDIs all seem to cluster together. So then what we did is we used MDI since it seemed to be very useful. And I'm just going to show you the therapeutic study. But here we look at MDI, and that's represented by the y-axis. And so it's negative down to the bottom, and it's positive down to the top. And the positive means that you're going to be having more of those ones that are associated with H. pylori infection, those 2 that were associated with H. pylori infection, and less of the 8 that were tended to go down when you have H. pylori infection. And so we can see if you have only H. pylori, that we do indeed have the MDIs are all very high in those samples. But interestingly, with the empty L. lactis, even though the L. lactis did increase the raw numbers of genera detected by 16S sequencing, when we look at the MDI specifically, the empty L. lactis did not seem to help with that. However, the AMP expressing L. lactis and the GAMP expressing L. lactis, they both were able to give us a nice low MDI. And also antibiotics gave us a lower MDI as well. So apparently somehow the antibiotics were specifically going after the bacteria that were associated with H. pylori. So summary here is Acinetobacter and Staphylococcus were positively correlated with H. pylori infection. We had 8 other genera that were negatively correlated. And MDI was able to predict taxonomic assemblages associated with infection by H. pylori. And we also had the guided AMP, and antibiotic treatments were associated with low MDI. So the conclusions of, total conclusions from our studies is that the MM1 guide peptide confers a high specificity, and it's very useful that way, against H. pylori. Probiotic guided AMPs eliminate H. pylori in mouse within 5 days with just a single dose. And they also work very powerfully as a prophylactic as well. And the elimination of H. pylori, when you eliminate H. pylori, you really tend to have a big increase in microbial diversity. H. pylori just causes a lot of devastation, and if you get rid of it, we get increases in microbial diversity. Guided AMPs did outperform AMPs in the long term in prophylactic study in terms of preserving diversity. So we want to really go after that and study it more. And then finally, the final conclusion here is, well, probiotic GAMPs may turn out to be a superior cost competitive replacement for antibiotics for H. pylori control. And so there may be an avenue for engineered probiotics. And if so, I think this would be an excellent candidate for that sort of route. Well, that's the end of my presentation, and thank you for your attention.